And okay, can everyone see my screen? Okay, so even though the title of this is Deep Learning and Computer Vision Will Transform Entomology, I'm not even sure where how I ran into this archive manuscript that's pending publication in PNAS. But I immediately contacted Toke and said, hey, we should get together, talk about sharing resources and convolutional neural net algorithms, et cetera, et cetera. And the more I learned about what he was doing, I was just fairly amazed. And so he's actually hit on what I think are three really, really sort of key areas that I'm interested in. And so I don't view this as a one-time presentation. I'm really hoping that one or more working groups take hold after this and get some collaborations going. And so the three areas that I want to follow up on are obviously the computer vision, artificial intelligence, like segmenting images, individual images of specimens from a group of specimens, sharing images and data and various codes, scripts. But what Toke and his colleagues have done is, I just think is amazing, is embedding this computer vision and AI into these monitoring instruments. And you know, we've been talking about this. He's the first person I've seen that really has delivered on these. So I think he's going to talk about BioDiscover. That's where you just pop specimens in the hopper and you get area, you get identification, blah, blah, blah. And they've also developed an automated light trap monitor, primarily for moths, and what I call a pollinator monitor that you can use not only to look at insect flower visitors, but also flowering phenology, et cetera. And with regard to that, I think there's a lot of other things beyond insects, and even plants that you can use these for. And then one thing I want to do is link collections with inventory and monitoring efforts. And he and his colleagues, once again, have done a lot of amazing work in monitoring in the Arctic, especially this one area in Greenland that I think he'll talk about. And that's where he wants to deploy a lot of these technologies. So I'm really looking at trying to develop coordination among countries and continents. For a long time, I've been pushing this, trying to get US researchers interested in the new Arctic. And I think Toke offers an opportunity to really move that forward. I'm also interested in the tropics, and I think a lot of these things could be deployed in areas of the tropics that we can't really access very easily. And so hopefully among this, we'll share designs that, you know, maybe they came up with a design in Europe and we find somebody in the US to do the 3D printing of the designs or deploy prototypes in the field to test them. But hopefully we can target some joint funding resources. And with that, I'm going to stop sharing and I'll let Toke share his screen. And I'll just be admitting people. So Toke, can you?
Yes, I think you might be able to see my screen now.
Yes.
And you can hear me all right?
Yep.
Okay. Do you want to admit more people, or could I just go ahead now and start?
You go ahead.
Okay, well, first of all, thanks, Neil, for reaching out and for organizing this.
online presentation. I'm really grateful that you gave me that opportunity, even though you sound as if you were the one excited. I'm actually also quite excited about having the opportunity to reach out to particularly North American colleagues. I think we haven't interacted so much yet, so I'm really looking forward to that. Basically, what I'm going to say today is what's in the PNAS perspectives paper that Neil talked about, plus some more. And I do have this Twitter ban symbol here, but it's actually not because I'm particularly concerned about sharing these ideas and preliminary results. It's really just to adhere to the embargo of PNAS. So understand that I'm really open to collaboration and to sharing ideas. So if you have any questions later during the presentation or afterwards, do reach out. I'm sure we haven't found the best solutions for all of the things we've tried to get a start on. So yeah, a few words about myself. I'm actually just an ordinary ecologist that have worked for quite a while in ecological monitoring, particularly for insects, but also for plants and vertebrates. And over the years, I've realized that we do things in a very labor-intensive way as biologists. And I've been fortunate to work in the last couple of years, 3 or 4 years, with software engineers and electronic engineers to help kind of transition to a state where we are maybe able to get solutions for automation of some of the tasks that we as biologists have been doing for years and years or decades and decades. So I'm going to start a little bit broad and focus in on entomology as I go along. And I hope to spend a little over half an hour, so there will be a little bit of time for questions afterwards. So I was quite inspired by a paper that came out in Nature Ecology and Evolution a couple of years ago. Does this make a difference for you so you can see my entire screen?
Anyway. I can see it.
You can see the entire screen. Okay, it's just that your faces are covering part of my screen here. Okay, so this paper by Estes et al. Kind of revisited old ideas by Simon Levin from 1992 that claimed back then that actually we as ecologists tend to try to make inference at scales at which we can't really observe ecological phenomena. So we don't really have the tools to observe ecological phenomena at the spatial and temporal scales at which they happen. And Estes et al. Tried to ask that question again some 25 years later to figure out if things had changed, and their conclusion was that actually when it comes to the scales in space on this panel and in time on the y-axis, we are actually still using rather conventional field techniques. So we have all these remote sensing techniques, but ecology in general takes place in this lower left corner, and entomology, if anything, takes place in the very small part of this lower left corner. So we still need these new tools to study ecology at the scales at which ecology really happens. And image-based solutions might offer a new way of improving the resolution in space and time for such ecological studies. Here I'm obviously not showing you entomology, but the cover of PNAS some couple of years ago that presented a really nice study on image recognition of Savannah wildlife. And it's one example where game cameras have been used to count and identify mammals on the African savanna. And the panel to the right here shows you bar charts of the number of papers after the 21st century that have used artificial neural networks to process image-based data for describing organisms, counting them, and classifying them or identifying them. And even though you might think that these trends are on the rise, if you compare this to how machine learning is used in other disciplines, actually in ecology, we are only scratching the surface. This has really taken off in other disciplines, but in ecology in general, and I would say in entomology in particular, we're still not really implementing these tools. Anyway, I can recommend the review by Ben Weinstein in Journal of Animal Ecology as a primer for this argument. Some of the work that I've been doing in the past relates mostly to seasonal dynamics of plants and animals, phenology. And some of our results show, for instance, that if we study phenology across different plots in a landscape and through time, we can, by spending hundreds and hundreds of hours in the field, we can detect patterns like what we see here on the left and in the center, that flowering seasons.
Toke?
Yes.
Are you the one that's typing?
No.
Whoever is typing, can they stop? Or can everybody but Toke mute themselves?
Right. Yeah, that's better.
Thanks.
Okay. Yeah, so these panels show how we, based on manually observed data, really hard-won data, can demonstrate that flowering seasons in Arctic and alpine environments are becoming more synchronized. They are kind of advancing more towards the end of the flowering season than in the beginning of the flowering season. My point of showing you this is that this was really hard to detect because there's such interannual variation in flowering times that this really suffered from the low temporal resolution of phenology data. And phenology data is typically something like this, where you have multiple repeat visits to the same location, and you count either the number of individual insects or individual flowers, and you derive some seasonality curve. But if you have only a few visits during the peak period of activity, then something like detecting changes in the duration of this activity period is really hampered by the temporal resolution of your observations. So with much more frequent observations, it would be much easier to detect any changes to the duration or the shape of such phenological curves. So that's one case in point where I think if we increase the temporal resolution of our data, we can make new inference also of relevance to the interactions among organisms. Similarly, when it comes to insects, we often kind of trade the time we spend on individual plots against the number of plots that we can actually visit. So here's a study from Svalbard where friends of mine spent 31 full days on the cold Arctic, high Arctic tundra observing flowers for insect visitation. And what they were able to observe were 263 insect observations over the course of 31 full days of observation, where they every 10 minutes shifted from one plot to the next plot to make sure they had good spatial coverage. But you could argue that that's really hard-won data, and 263 insect observations could maybe have been achieved by more automated methods. I'll come back to that. So as part of a project we started a few years back, we began to implement these modified wildlife cameras that record on a fixed time schedule. So they are time-lapse cameras. We mounted these cameras over patches of a particular Arctic flowering plant, the Dryas octopetala, that some of you may know. It's down here on the left. And we did this across the landscapes at different field stations, but also across different sites in the Arctic. And we were interested both in understanding this fine-grained species interaction data between the plants and the pollinator, but we were also interested in addressing this in the context of the species pool of pollinators at the different sites. So we looked at the same flowering plant species in context where they had access to different pollinator species. And we were hoping that by using the cameras, we could also track the destiny of that particular flower that we monitored with the cameras through to the stage where it would set seeds. So I'm going to, yeah, so first of all, here's the map of those different sites where we did this work. Neil mentioned a site that I've been working particularly intensely at. That's the Sackenberg Research Station here in Northeast Greenland. But we teamed up with colleagues that also work at other sites in Greenland and in Fennoscandia and on Svalbard and Iceland to be able to implement the same kind of observational setup at multiple sites. And these sites differ in mean annual temperature, but also in the diversity of the pollinators. You could argue that this is particularly relevant in the Arctic because of the climate change, but also because the vegetation is more two-dimensional in structure, so it's easier to monitor the plants and the pollinators from above. But yeah, we can come back to the limitations of that. So here's a small video sequence that shows you some of the images that we collected with this camera setup. And it's really just a video that stitches the time-lapse images together. You can see the flowers as they move through one day. In the lower right corner, I think you can get the time of day. And you can also see that some of these flowers are visited by insects. There was some flickering in some of these flowers, and that was the syrphid flies mostly that visited these flowers at that particular day. Now it loops again, and you can see more of this these insect visits. So as a starting point for using artificial intelligence to detect both the flowers and the insects, we manually went through these images and scored the phenological stage of each of the flowers and recorded whenever a flower was visited by an insect. And if you imagine that data kind of superimposed on this image, it looks something like this, where you have in the same XY coordinate space, you have here colored dots for records of the stages of individual flowers, and black dots are the individual insect visits to those particular flowers. So you can maybe see one of those clusters of points here is a flower that moves a little bit back and forth in space and shifts through its different phenological stages, and we record each of the insect visits to that particular flower. But from this panel, you can also see that there are some flowers that are only visited by very few insects, like over here, and there are also some flowers that didn't have any visits at all. So I think this gives you maybe a feel for what realm of data that we can actually look into if we would be able to translate image-based data on plant pollinator interactions into more kind of standard occurrence data and phenology data. Now, if you can imagine putting this data on a time axis so that each of these individual flowers recorded as a kind of life cycle graph, that looks something like this top panel here, where you have time on the x-axis, and each row in the panel is one individual flower. Most of the flowers, when we started this video sequence, they were in the bud stage and then transitioned into open flowers, became visited by different insect species on a few days, and then eventually wilted. And some of these flowers eventually produced seed capsules. That's the blue stage here. So if you imagine that we can track individual flowers through time, we can really start addressing questions of how the interaction between plants and pollinators translate into the probability that the individual flower produces seeds. So we can link interactions to ecological function, if you like. We can also see that even from this one video sequence of one particular patch of flowers, we can actually derive rather detailed information about the diurnal variation in insect visits. Maybe not surprising that there was the highest activity at the middle of the day, but this gives us a quantification of how that looked for this particular site, and we can compare that to other sites or other years. Similarly, we got information about when in the season we got most of these insect visits. From just a 10-day sequence of images, we can clearly see interesting ecological patterns arise. Some of this, in this particular case, obviously relates to the number of flowers that were actually available and offering resources to insects. But it's kind of as, a glimpse into what possibilities these image-based methods could offer. And the next stage for us was for the flower detection to train a neural network based on these manually annotated flowers in different stages. And I have a video here that shows you what that looks like. The prediction of this network is formatted in a way that anything the neural network does not recognize as the object of interest, the flowers, is grayscaled, and everything that it does recognize as the object of interest is in color. So if you look at this video, everything is gray in the beginning, and as these flowers begin to open up, they are colored, and I would argue that almost all the flowers that are open, they are colored and therefore detected by this neural network. So this means that we can actually automatically feed the neural network these images, and it will count and position each of the individual flowers on our scene, so we can potentially even track the individual flowers and relate the number and timing and diversity of insect visits to that flower to the function of in the form of probability of seed set later. Okay, so now I'm gonna show you one of the images behind this video. Looks something like this. Some of you might know about confidence values. That's the typical way of neural networks to tell how certain the network is that the object is the particular class that it's supposed to detect. And these values come between 0 and 1. So you can see values that are really close to 1 here. That means that the network is very certain that what it has detected are in fact flowers. And it also looks rather convincing that it is able to detect these flowers. So we're obviously excited about this from a plant perspective, but I think it also offers great opportunity for plant pollinator interaction studies. And when we compare some of these individual time series, we have now hundreds of these, but these are just some example ones where we have the ground truth data. We've actually gone through the images by hand, if you like, or by eye, and recorded the number of flowers in each of the images. And here the ground truth data are in blue and the detections are in red. So we have fairly good congruency. It means that we can trust this neural network in its ability to detect these flowers. It's not perfect, and I'm not showing you all the details, but at least you can maybe get the point that that for this particular flower species, it is actually possible to feed these raw images and get phenology data back. We are working now with a platform called Suniverse to implement a citizen science-based evaluation of whether our flowers have insect visitors or not. And this is right now in a beta test phase where volunteers are trying out our task and evaluating whether they think it's relevant, too difficult, too easy, or whether there's anything that can be improved. I think this platform could be used not just for this particular case study, but for many other studies related to insects, and could be of great use in our kind of future efforts to implement these machine learning techniques. I am going to present a few other things that we've done with respect to field tools, and then I'll move into the lab-based tools and end with some conclusions. So here is some images that are actually also in the PNS perspective that exemplify how we can move even further with these camera-based solutions. So in this case, we are not using an off-the-shelf wildlife camera, but we've actually built our own more intelligent camera. It's based on mini computers, like you may know Raspberry Pis. There are also now mini computers that even have image processing capabilities. What we have down here in the corner is what's called a Jetson Nano computer. It's a very cheap computer, but it actually is able to process the images as they're recorded. So we can record images at really high frequency, not just the minute frequency that we had in the previous video, but down to maybe 1 image per second. And rather than storing all these images, the mini computer can actually process these images on the fly and detect whether there are insects on the images or not. That kind of information translate into, when we have cameras over an entire season, really detailed time series for individual insect species. Here for the honeybee and species of bumblebee and a species of syrphid flies, which were the dominant species in this particular flower bed. But we also get this detailed information about diurnal activity. And as you can see up here in the top right, once we have this functionality in the hardware, we can even think about ways where we don't necessarily need to store and ship all the images to a central server, but can just ship the XY coordinates and the identity of the detected organisms that we saw in the images. Obviously, we need to store some of the raw data as well. That can be done on SSD disks like this, but it means we're approaching a real-time solution for insect monitoring, which in some cases can be super important, not just fun. So I'm going to show you another video here that shows how this tracking algorithm is implemented on this particular flower bed. Here you see a video where the individual bumblebees are actually tracked on images taken only 3 seconds apart. It also means that we can start addressing more behavioral questions of even interactions among pollinators and their relationship to different pollinator or different floral resources. So in this particular panel, you see again one of the images similar to what you just saw, but for these 3 different species that you also saw in the previous slides, here are dot patterns of where in those images were the individual species actually recorded. And what's clear here is that the honeybee and the bumblebee, they exploit the same resource, these purple flowers, whereas the syrphid fly, based on these image recognition tools, we can demonstrate that they are actually not visiting the same flowers, even at this very local scale. So we can start asking questions about niche segregation and resource partitioning with these new tools. Okay, so I also want to show you a little bit about this Moth Trap, which is built on the same philosophy of this pollinator tracking device, that it's basically a computer and a web camera that's built into this box, but it's constructed over a scaffold where we have a UV light and also a ring flash to make sure that we can record moths that get attracted to the UV light and land on the screen at night. But not only does this device record the images of the moths, it's also tracking the individual specimens so that we can avoid or at least minimize double counting. And having processed these images, we actually get, again, seasonality data, much like we got for the plants and the other invertebrates. Here for individual species of moths that were trained for this particular setup. This is something that once it's running, the images can be recorded even before the neural network is completely up to speed with the classification. So I would say this is maybe a starting point for something that can be implemented across biomes and countries, certainly. Yeah, so overall, I think these field methodologies, they are really valuable for automating phenology observations, but they also offer a way into more functional ecology of understanding something like thermal performance of organisms that are not the typical lab organisms like Drosophila or other favored model organisms. Here we can actually study how organisms respond to microclimatic conditions and variation in situ in their natural environment, which I think is lacking. We can certainly also start looking into species interactions and particularly for remote areas, but I think also for global comparisons, it offers a new opportunity for ecological monitoring. So yeah, there are other resources that these tools can be implemented over. We are doing some work now monitoring the fauna of dung pads, and we're also working with Norwegians to monitor the beetle fauna on polypores. That's this lower right image. But it goes beyond that. We can also now monitor invasive plant species or roadkills using camera techniques, and for wildlife studies, we do a study with an Italian group about the relationship between rodeo and humans based on computer vision techniques on wildlife cameras. And we are only just beginning to, but I hope to work more on the implementation of standard cameras with thermal cameras, so that we can get into more detail about the microclimatic response of arthropod behavior. So that's for the field applications. In the lab, I promised to say a little bit about this biodiscover device. That is actually out in a separate paper in Methods in Ecology and Evolution, where we describe both the hardware setup. This is basically a couple of cameras, industrial grade cameras, and a light source that records images of a specimen that's dumped through this funnel into an ethanol-filled glass or quartz cuvette, which offers a very good environment for high resolution images of specimens at macro scale. I've got this example here of the ladybird beetle that is, here it's animated, but in reality, you should think of this as one specimen that gets dropped through this cuvette, and the tracking algorithm only crops out that part of the scene of the camera that shows deviation from the background. So we only store the information of the specimen as it swirls around and goes through this ethanol-filled column. Maybe I didn't bring that. So the idea is that after one specimen is imaged, we can control whether it lands in a separate vial from the previous specimen, or it gets mixed with the previous specimen. So in this way, we can actually implement a sorting functionality on top of this classification application. This is again a table from that paper, but it's a classical way of describing the performance of a classification algorithm, where the known identity of these different species is given as rows, and the predicted identity of the same specimens is given as columns. And if the classification routine is perfect, then every specimen would land on the diagonal and the value would be 1, which is the case for some of these species, whereas others have values that are slightly below 1. It means that there are some misclassifications. But generally, across this test set of taxa, where we knew the identity of spiders and beetles and some flies, we got a very high overall accuracy. And in those cases where accuracy was not as great, like this one beetle species, we could actually relate that to a fairly low sample size. So had we had a bigger sample size, probably the performance would have been even better. What these images also allow us to do, because the computer vision that's implemented in this system automatically computes a series of geometric features of the specimen in each image on the fly. So it's done while the specimen actually sinks through the ethanol, is, for instance, area and circumference and diameter, different geometric features. One of them, the most simple one, is just the area of the specimen in a two-dimensional image. And we've taken the effort to subsequently dry and weigh individual specimens of, in this case, Danish wild bees. And you can see how close the relationship is between the area of each of these specimens. That's the mean of all the images of that specimen and the dry weight of that particular specimen. So we argue that we have a fairly good and certainly rapid tool to estimate biomass across species. So as we generate more data like this, I think we have a very nice opportunity for also estimating biomass from insect samples non-destructively. Yeah, this is maybe not, yeah, for some of you maybe relevant, and for others maybe not so much. I just wanted to bring this study that we did last year on specimen from a museum collection. This is the crabbit beetle collection from the Natural History Museum in London that one of my students scanned the entire collection with help from obviously from the collaborators at the museum. And he cut out the individual images of each of those 65,000 specimens, and we grouped them according to species and asked the computer how well it would classify these 291 species to the species level, but also to genus level. And the point of this paper is really that if you are more concerned about accuracy than more concerned about classification accuracy than taxonomic precision, then maybe you can trade taxonomic precision against accuracy by saying that I only want to know which genus this specimen belongs to. Then you're more often correct than if you really want to go down to the species level. So I think that has some general implications as well as for how we use classification algorithms in the future. So my overall take-home messages is that we really need more high-resolution spatial temporal data, and that goes not just for occurrence data, but also for interaction data, and image-based tools offer a solution for that. And this goes particularly for entomological work. Time-lapse cameras and this combination with deep learning really opens the door into new types of data, particularly when it concerns the interaction among individuals. And we can go from the population level down to the individual level, which I think is really exciting and relevant. We can do collaborative projects where we even ask people that have less insight into the details of our study question to deploy cameras, obviously following protocols, but this is a way that we can get comparable data across multiple sites. And I think also in the lab, these tools I've shown you can open up new avenues for entomological research. And I'm sure you have your own ideas of how these tools can be implemented for your questions. And I'd be happy to discuss ways that we can work together to reach some of those goals. So we've got some collaborators, mostly at Aarhus University, but particularly also in Finland. And well, they're coming up now in the UK and in Germany. So this is a family that's growing. We've got funding from a private Danish foundation, the Research Council of Denmark, and from Horizon 2020, the European funding agency. Okay, I think that's all for now. I'll stop sharing, and I'm happy to take any questions.
Oh, Toke, before go, or maybe you don't need to share. Could you talk a little bit about the DNA barcoding option for BioDiscover?
I can. I don't think I necessarily need the slides for that. We talked a little bit about that before, Neil. This is not really something we have done any major work on yet, but we are speaking with molecular biologists about really combining the robotics components of the BioDiscover setup with molecular robots that do DNA extraction and PCR, so that we can ultimately actually fully automate the process from bulk sample via biomass estimation and maybe a sorting of specimens of particular interest for maybe taxonomic inspection or or further study from those that we can sacrifice and use for DNA extraction. So I think there's certainly merit. I'm not sure there's a clear path described yet for how to merge the image-based and molecular-based identification tools, but we are certainly, we've started that discussion.
Okay. Are there any questions?
I had a quick question. First of all, super excited to hear about all this work, and I'm really lucky to get to see it kind of in real time as it evolves. My question was, you're using these images at a fixed scale to calculate kind of the area and biomass of these specimens, but how would you go about calculating area for images that range in scale? So they're from different sources, like, you know, the community science images on iNaturalist, or the images in bold, which have absolutely no ruler, or, you know, the museum specimens that could have a ruler or a centimeter scale or a 5mm scale. Like how would you approach that problem? Is that kind of the next, is that on your kind of next things to tackle?
I am not sure, actually. So we've only used the biomass estimation for images that were recorded with the BioDiscover setup. And for that setup, we know the exact distance from the camera to the cuvette. So it is on the same, all the images are recorded on the same spatial scale. You're absolutely right that when it comes to more randomly or haphazardly collected image data, there is no scale. And I would say the only thing that I can think of right now is that if you know this, if you can use the images to record the species, and you have an independent information on the size or the biomass of that species as a mean, then maybe you can translate. But I've actually not given that much thought. We were only concerned about deriving biomass estimates from the images that are recorded with the BioDiscover machine.
Right, thank you.
Talking about like the transferability of some of your learned classifiers. I mean, I guess you could start with the first one you told us about, where you had the multiple yellow flowers in the image.
I'm not sure I got the first part of your question. There was a little bit of echo.
Okay, let me stop my video just in case. Would you mind talking about the transferability of some of your learning systems? Like for the first part of your talk, you talked about, you know, multiple flowers in the image and being able to automatically segment out the flowers and also be able to find the insects. Did you use multiple cameras and multiple setups for that, or was that all just done on the one single camera?
So yeah, transferability. I think that's a great point. We for the work on dryers, we have multiple cameras, but they are all recording different patches of ground. So we don't have any stereo approaches, if that's what you're thinking, where we have two cameras pointing at the same objects.
Mostly, I just meant when you trained the classifier, did you use data from multiple cameras or just all the same camera? And then could you then use that on a new camera?
So in in the work we are currently writing up on the dryas flowers, we are training a model based on a subset of cameras and testing the performance on images from different cameras, but still recording images of the same plant species, maybe in another site, maybe in another year at the same site, but different cameras and kind of independent data, but all on the same flowering plant species. So in terms of transferability, we couldn't use that same model to detect a very different type of flower. We know we sometimes detect other species of or flowers of other plant species that occur in the same scenes as we have our focal plant, but in a way that's that was unintended. And I think as we move forward for different flowers of different morphology, we'll probably have to work our way through this data one species at a time. When it comes to the pollinator detection algorithms that I showed in the center of my talk, where we didn't actually first detect the flowers and then flowers with or without pollinators, we have experimented a little bit with transferability, where we have images recorded over a particular flower bed, like these succulent plants you might have seen in some of the videos of the genus Sedum. And we've used those also, the models trained on those data for pollinators to potentially detect pollinators over other plant species. And while that works to some extent, we get a lot of false positives. So a lot of objects in these new plant species gets detected as potential insects. So one of the challenges that we are working on right now is figuring out a way that would be helpful to the ecologist and not be not violate the assumptions of using algorithms on unknown data. There is kind of a trade-off there between making a rigorous evaluation of performance on the one side, and also just getting the job done of actually detecting some of the insects in a new series of data, so that we know where to go and search manually through the images that we think are most likely to contain insect visits. So you have to think about this, that the pile of images is so big that we cannot just go through the entirety of our collection of images. We have to have some pointers as to where would we be most likely to find training data that can be used to refine our detection algorithms. So maybe that's yeah, kind of answering your questions.
Yeah, that was great. Thank you.
So Toke, there's a question about the size of tubing, and I thought you told me that you were developing a larger cuvette for larger species.
Yeah.
Are those replaceable within the unit? So can you just pop out a smaller size cuvette and put in a larger size, or how does that work?
Well, to be honest, we are not that far yet. We have a system, that's what's described in the MEE paper, where we use a 1 by 1 centimeter cuvette, and that's fine for some insects, but obviously not for many other larger carabids or bumblebees. And as I told you earlier, we are working on a separate setup with a 3 by 3 centimeter cuvette. Now you have to think about this as there are several different challenges. One of them is fitting the insect into the cuvette. That's rather simple, and you could imagine that you could simply just replace the cuvette. But if you also want to control where that specimen goes after it's been imaged, then that's where the challenges starts to pile up, because in the original version developed by my Finnish collaborators, you would have to empty the cuvette for each specimen that you had imaged, because there would not be an opening in the bottom. So you have to pick out the cuvette and empty it into a tray or some other place. And we found that to be inefficient in the kind of grand scheme of things. So we are working with a flushing mechanism, and that flushing mechanism would depend on the size of this cuvette. So that's why it's not quite as straightforward for us to just replace the small cuvette with a larger one, because the flushing mechanism also needs to move fast enough that the ethanol in the cuvette doesn't go all over the place, but actually lands where we want it to land. So we are working on this, but it's we are not there yet.
Got it. And so this prototype BioDiscover, I thought you said it cost you about 8,000 to put together.
Yeah, well, I think it's maybe even a little less than that. But to be honest, we are only making a few copies of it now to some close collaborators where we know the long-term usefulness for their monitoring purposes, because we're not really a company that can just mass produce these. So even though they are mass producible, we've been a little bit reluctant to just make a web shop at this stage. But I think eventually we will rather provide the drawings and the possibilities for others to build them on their own. And now we're a little bit in the in-between stage where it's not that easy to build from scratch, even if you have engineering expertise. So we will be working on ways that this can kind of be made easier for collaborators in the coming time.
Okay, thanks.
I've got a question for you. Great talk. Very interesting work. I wonder if you could speak a bit about kind of maybe the training sets that go into when you're trying to delimit morphologically similar, closely related species, like in the carabids, or even telling bombus from a syrphid, in the sedum plots. How big are those training sets, and have you evaluated kind of their efficacy for finding new stuff if you happen to get a different syrphid coming into those plots?
We certainly do see other species of syrphid flies in the images, and there are some errors in our detections. We can see that from our performance evaluation, but we can also see it from our predictions. And right now we're working on ways to make that visible to the human eye. It's a little bit the same as I said before. We have millions and millions of images, and we cannot just scroll through all of them. So we have to find ways that the details we're interested in become manageable in size. And one of the ways we're working with this problem is by actually by letting the neural network tell us where it finds its detections, and then crop the images around those detections. They are basically a square box in an image where the network thinks, here is one of our syrphid flies. Then we can run scripts to crop out all these detections as small individual images, and those you can actually view as thumbnails in a photo editor. And there it becomes often quite clear that, say, 80% of these or 90% of these are in fact that one species that we thought it was, but that in between those 80 or 90%, there are maybe other species of syrphid flies or beetles we hadn't even thought were there, or slugs or ants or something else. So right now, this is an ongoing dialogue with the engineers, because they are not really used to this kind of diversity of classes. And although there are ways, and we discussed that a little bit in the perspectives paper, for detecting unseen classes, or classes that the classifier doesn't know about. This is really an area of research also in computer vision engineering. Yeah, so I guess maybe that answered some of the question.
Yeah. So it's not necessarily, going out and saying we only see, or, most of the time we see these 3 species. So then kind of creating a training set for the diagnostics of those. It's more kind of focusing on the neural network to be able to pick out the bugs and then whittling it down to a more manageable kind of chunk there.
Yeah, well, I think they are related but not mutually exclusive strategies. One could be that you just want to detect all the insects that are there, and then later worry about which identity they have. Or you pick out some morphologically distinct classes, and then you ask the network also to show the cases where actually the confidence values are low, so the network wasn't even sure that this was the correct species. And they may be examples of that new species that you hadn't seen enough examples of, that you could train a new classifier where that is included. So I think we are probably looking at some iterative processes where either citizen science can help us identify these new classes, or we kind of retrain networks that become smarter and smarter with time. Again, a reason for working together.
Very cool. Thank you. And sorry for the unmuted sneeze in the middle of your talk.
Oh,
Well, and so you've only primarily talked about visual, but this really can be applied to things like sonar. So, for example, you could detect mosquito species based on their buzzing. And I could even see by the end of the decade, we could put sensors out in the field and detect leafhoppers chirping.
Yeah, we also have a paragraph about other sensors like acoustic sensors, or even the work that's done on using radars. And the way we phrase it in the paper is that they are certainly complementary, and you can imagine that in the future, maybe you combine these sensors so that that you have both some optical and acoustic signal. One of the challenges with acoustic and radar signals is that it's not that easy for the human perception to evaluate if a particular signal was in fact mosquito species A or B. So it's very hard to kind of use independent data to validate the signals that we acquire, whereas images they're kind of more intuitive to us. So we can present an image of some insect to 10 experts, and if they all agree that this is probably that one species, then we have independent confidence that it probably was that species. So that I think is a difference between the acoustic and radar and optical solutions.
And I started to think about all the different trap types that I've used. And for all the ones I could think about, I could think of modifying the design to fit this. So like a Malaise trap has a cylinder at the top. Well, if you replace that with a collector that was more like an ant nest colony thing, so the insects would spread out, but they'd still be contained, and you could image what you captured from a Malaise trap. You could do the same thing with sticky traps. You could actually, I would be interested in putting cameras in front of pitfall traps. So I could not think of a trap that I couldn't use this technology.
Yeah, from my own history of going into this field actually started by putting cameras over pitfall traps. That's how it started for me. I was really curious to figure out when, during that week when I didn't look at my pitfall trap, were the trapping events actually happening. And by chance, we realized that some of the flowers next to the pitfall trap actually also had insect visitors. And that's how it started. Then we realized, oh, here's some independent data we can collect using these wildlife cameras. So, yeah.
Okay, and I just realized you should actually be going.
Yeah, well, theoretically, I've got a few more minutes if there's a question or two at the end. But I mean, otherwise, I'm clearly interested in continuing this discussion in a different meeting, or in working groups that are more tied to more specific topics. Anything, or if you have some individual questions that you just want to send me, I guess.
And I put the link to the Google Doc up there that people can access even after the webinar. And yeah, you can also email me and just let me know if you're interested in pursuing this, because like I said, this is not a one-off. I'm really hoping that we get some working groups coming out of this.
Yeah, I just put my email address in there in case you didn't have it. But yeah, no, let's make this a group group discussion if at all relevant.
Yeah.
If you have a minute, I'm interested in hearing how you managed to get the involvement of the experts in computer vision and machine learning and things in these efforts.
Yeah, no, that's a good question, and actually not a trivial one. I think
Yeah, because they are some of the most in-demand people on the planet. So
yeah, well, I shopped around a little bit in the beginning. I have to admit that we were really keen to develop what's now the BioDiscover machine, and I spoke to an engineer who did artificial insemination of pigs, and some engineers that work very closely with industry to automate assembly processes for companies. And they showed some interest, but never really fell in love with the idea of working with insects. So I think it took a couple of iterations before I landed with the collaborators that I now work quite closely with. So I guess it's like with any other collaboration, that there has to be this both the mutual interest and the trust and the long-term commitment. I think that was really my key criteria, that it would be something for the longer term, where we are not just collaborators on a single project, but more on a vision. And I think this new perspectives paper is kind of cemented that relationship, because we are really writing this together with both the signal processing experts and the mechanical engineers and those that have a specific expertise in neural network architecture and computer vision. So I would encourage you all to look for such collaborators, but put some time aside for it, because it might not be the first person you speak to that will be the long-term companion.
What do those people get out of the collaboration?
I think they get real world challenges. That's at least what the students that now that this collaboration is also through joint students, and the students that we co-supervise, they are often excited about you could say the UN sustainability goals, or the grand scheme of things, the biodiversity crisis. They want to contribute to solve a societal challenge. And I think that goes for the supervisors as well, to some extent. But actually, with respect to some of the academic challenges for them, biology or ecology actually represents a particularly hard challenge as well. The number of classes for our insect samples is much greater than the classes they work with in driverless cars or in other tasks. And for plants, the fact that the class changes through time, that the plant actually changes shape and morphology, that's also actually a real pure research challenge for them.
Great, thanks.
Okay, are there any other questions? Well, I think that does it for this. I'm sure we'll have another meeting in January or February and continue this. But thanks a lot, Toke. That was a great presentation.
Thanks.
Okay.
Thanks for organizing.
Okay. Goodbye, everybody.